So the competition that we're going to be talking about today, we'll get to that in a second. Don't worry, we've got a slide dedicated to that, is the Human Protein Atlas image classification. And we're going to start off by introducing a broad perspective of the competition itself. Then we're going to talk about the data that was provided for it. We're going to move on to talking about the way that the competition was scored, so how they evaluated all the models that were built for it. Then we're going to talk about the different kind of models that people built, just generally, to begin with, and then more specifically our solution, followed by the top solutions. So I think we have first, third, and eighth in there for this competition. Then we're going to finish up by talking about what we learned. But before we get into any of that, we're going to introduce ourselves. So I'm David. I have a PhD in theoretical chemistry, and I'm also a former statistician from the Bureau of Statistics. And I'm currently looking for work. Although, if anyone here is hiring, I won't be able to talk to you for the next few weeks, because right after this, I'm heading off to Australia. And as far as editors go, I tend to use Emacs. I'm very old school in that way.
I'm Jordan. Is this working? Is it working? Okay.
You got the bad one.
I'm currently a full stack web developer, but in May I'm going to go back to school to try to get a computer science bachelor's degree, and then hopefully afterwards get a master's with a focus in machine learning. And for editors, I mainly use Atom, but at work I use Visual Studio, but I don't like it very much. I like using the terminal, and I don't like clicking buttons, and Visual Studio makes you click buttons instead of typing things. Yeah.
Okay. So the competition that we participated in together recently was, as I said, the Human Protein Atlas image classification. So the goal of the competition was to build a model which could identify where proteins were located within cells based upon fluorescently dyed microscope images. So it was a fairly well-funded competition. There was a total of about $37,000 worth of prizes, 14,000 of which went to the first place. And it was hosted by the Human Protein Atlas, which is basically a data bank in Sweden which has a whole array of these kinds of images. So microscope images which have been fluorescently dyed. And the reason they were interested in submitting this as a competition was because those kinds of images are relatively easy to produce, but they're much more difficult to actually interpret. So you normally need a specialist, somebody with a specialization directly in interpreting these images. And a lot of the time, well, I mean, it's very similar to interpreting an MRI, for instance. So if this could be automated, it could save a lot of labor in that sense. I wanted to talk a little bit about, yeah.
I guess, why do they do that? Like, what is there to learn from these images?
I'm going to talk about that right away.
Are you?
Well, sort of right away. I'm going to talk about how they produce them first.
Okay.
If I haven't answered it after I go through that, then you can ask again. Yeah. So the way they produce these images is through something that's known as a green fluorescent protein, or variants thereof. So it's a fairly small protein. It's about 238 amino acids, and it looks basically like this. And it was first discovered in jellyfish. So we don't know exactly why they produce it, but if you expose them to UV light, then they glow green, like that. One of the things that's useful about it is that biologists discovered a while ago that basically you could take the gene for this, you could copy it, and then put it in another cell at the end of another gene for a protein. And then whenever that protein is produced in the cell, this dye will be produced with it, which means that you have something that's very easily visualizable to check whether or not that protein is being produced in the cell. So here are some examples. For instance, in the image on the left, you can see that pretty much all of the cells in the mice on the far left and the far right are producing whatever protein that this molecule has been bonded to, whereas the mice in the middle are not. What's more useful about it, though, is that not only can you tell whether or not the protein is being produced, but you can also tell where it's being produced. So in the image on the right, you can see that the protein that they're interested in is being produced by the cells in the feet and the beak of the chick there, but not in the rest of their body. And likewise, the chick on the right isn't producing them at all. You can also scale that down quite a bit. So if you view the cell under a microscope, then the bits that light up under UV light will be the parts that contain that protein. So you can tell where in the cell it is that this protein is going in the end. And that's extremely powerful if you want to figure out what the function of a protein is, for instance. Because usually if a protein does something, it will have to do it from a particular place in the cell. So it's a good starting point. Now, I think I might have just answered it then. Basically, the short of it is that if you can tell where it is in the cell that a protein is going, then you automatically get a lot of information about what it does, because certain parts of cells usually have a certain function, which means that, like, if you're trying to map out functions for proteins, then often the first step is figuring out, like, where it is that it actually ends up. So anyway, the images that were presented as part of this competition actually used 4 of these dyes. So they used a green one, which was which was attached to the protein that they were interested in locating, essentially. Then they used a red one, which was attached to microtubules, a blue one, which was attached to the nucleoplasm, and a yellow one, which was attached to the endoplasmic reticulum. And those last 3 were all used essentially as reference points. So they're elements that form like a structural part of the cell, so you can tell, like, roughly where in the cell you are by looking at where you are in relation to those. One of the interesting things that this meant, though, was that there were 4 channels for the input on this data set, as opposed to regular images, which normally have 3, red, green, and blue. And the channels also meant something very different, because ordinarily, if you're looking at an image, then there won't be pure red or pure green or pure blue at any point. So the combination of the 3 is normally what we use to describe some like color that's sort of in between them to a certain extent. So if you have a pixel which has a non-zero value for red and green, then that might mean that it's like orange or something like that. But in this competition, basically the images, if you had both red and green on the same pixel, then the only thing that would mean would be that you had both microtubules and the target protein in roughly the same place in the cell. So the interpretation of the actual channels is significantly different in this case. This also meant that the actual architectures of the models had to be modified a little bit if you wanted to use all 4 of the channels. So that was an approach that some people took, but some people in the competition decided to just drop one so they could keep the regular sort of image classification structure with the 3 channels as the input. And I'm not sure whether there was necessarily much advantage between the 2 of them. There was a bit of debate within the community about which one actually proved to be better, and even in the top place solutions, there was a bit of variety in terms of which ones people used.
Just briefly, before we go on, I wanted to mention, like, if we're thinking about why people dropped the cells and they found that it didn't change anything, like David said, that these were reference points, and usually people were dropping this yellow channel. And when we think about how, like, these cells, the morphology of these cells, the microtubules and the endoplasmic reticulum usually exist within the same location. So it could be that the endoplasmic reticulum wasn't actually providing any really additional data that couldn't be provided by the microtubules. So just kind of a theory that was out there. We don't really know for sure why it was doing that, but that's possibly a reason why.
And of course, by dropping a channel, that also means that your calculations are going to be a little bit quicker because you're not reading in as much data and you're not processing as much either. So in terms of the data that was actually provided for the competition, there were about 30,000 images in the training set. And when I say 30,000 images, I mean 30,000 microscope images. For each microscope image, there were 4 constituent images for each of these, so 1 for each of these channels, which meant that there was a total of about 120,000 files. By default, the images were at a resolution of 512 by 512 pixels, although the original images the original microscope images were also provided, and they were at a much higher resolution. So a mixture of 2048 by 2048 and 3072 by 3072. And a lot of the winning solutions did use those, but even they decided to scale those down a little bit because that's quite a lot of data they have in those. I think the total size of the data set was about 250 gigabytes. Yes?
Were the originals also 4-channel?
Repeat the question.
Were the originals also 4-channel?
Yes, so there were also the 4 different files for the originals. So, in terms of what actually needed to be discovered as part of the competition, so there were 28 different locations that were defined within the cells. So 28 different classes that the images could be considered as representing. And there was also a huge imbalance in terms of the number of images that contained an example from the classes. So for instance, about 13,000 of the microscope images had proteins that were located in the nucleoplasm, but only a couple of dozen had proteins that were located in the rods and rings. So huge imbalance there by something like a factor of 1,000 in terms of how well these classes are represented. It was also possible for the image to contain multiple different locations at the same time. So for instance, a good way of thinking of this is that proteins are usually produced in a particular part of the cell and then transported to that afterwards. So it's entirely possible that a protein could be located in 3 different places. And there were a lot of examples like that. So basically, the practical upshot of this was that this was a highly multi-class and multi-label problem with a lot of imbalance in terms of the representation of the classes in it. Kaggle, as part of the competition, also discovered that there was a lot of external data that they could use as part of this competition. So if you go onto the Human Protein Atlas database itself, then you can actually find about another 350,000 images on top of the 30,000 that were provided as part of the training set. Not all of those were labeled, though. So there are about 70,000 useful images that people found at the end of that. And to actually use those, you had to do a little bit of processing beforehand because they weren't presented in quite the same format as they were for the competition. But nonetheless, that represented a significant increase in terms of the amount of training data that was available for people. And this was something we only actually found out about after the competition.
Did they have to do a lot of work in getting the new images to line up with the competition goals? I noticed this one has some intensity ER objects.
Yeah, so the reason for that, this is viewed in a different sort of panel. These little boxes down here represent basically what you're trying to view in the image. So I think if you were trying to just get the data that they had for the competition, you would probably just look at the intensity, because that would correspond to how much they measured light from the fluorescence, I think.
Yeah, this was just an example on the website. So those boxes you could like click on and it would highlight different parts of it. So it was like a visual representation of these different filters that were available. So in the original data, there wasn't any of this like filter, like fluorescence intensity. There was just the image data. So you could actually download the extra data, but I don't know. I didn't really see many mentions of people actually using it, but it was just some interesting thing that this kind of like supplemental data to overlay over top of the images was available, as well as just the extra images with their labels.
People tended to just use the same sort of data that was available for like the images in the competition.
Yeah.
And I do have a question, just to make sure I'm completely following you guys. Can you go back one slide?
Sure.
So this is a multi-label task. So you're going to be given one image, and that image can have, you know, a nucleoplasm and a microtubule somewhere in that image, and then the value you give that image is like ones for those and zeros for everything else.
Yes.
Okay, and was there a lot of did most images have a bunch of different labels, or were they mostly?
I think if you look at the total counts for like the number of ones that were in all of the cells, it was somewhere around about 50,000 for 30,000 images in the training sets. So I think that works out to be a little bit less than 2 on average. So it was very common for there to be like at least a small number of like double examples. If that makes sense. Okay. So I'm going to hand over to Jordan now, and he's going to walk us through how the competition was scored, essentially.
So the submissions were evaluated on something called the macro F1 score, which is harmonic mean or weighted average of precision and recall. And precision, I put the formulas up there, but I'll explain a little bit kind of about what they actually mean in like plain English, which is just that precision is like if you have a set of results that are marked as positive, you want to know how many of them are really positive. And recall is like given a set of results that are marked positive, you want to know like what portion of those you actually missed, like how many of them were marked as falsely negative. And so why are we using an F1 score as an evaluation metric, and why do we use it in this competition? And why not just accuracy or just a mean of precision and recall? So to help illustrate that, I have an example, and we can kind of walk through the example, and it'll help paint a picture of what the F1 score does. So the example is 100 patients come in with warning signs of cancer, and out of 100 patients, one of them will actually have cancer. If you have a model that predicts nobody has cancer, it's 99% accurate, but it's not a good model in any sense. Like it's not going to serve the purpose that you want, which is to figure out who has cancer. But this model that predicts that no one has cancer has a precision and recall of 0, which means the mean of precision and recall would be 0. So to understand why we can't use the mean of precision and recall, we want to look at the opposite. So what if this model predicted that everybody had cancer? So it like did 99 false positives. The precision would be 0.01, and the recall would be 1, meaning that mean of precision and recall would be about 0.5, which would indicate like, oh, this might be a good baseline model, when in fact it's very far from that. So if we look at what's the F1 score for this model that predicts everybody has cancer, so when you plug those precision and recall into the formula there, you get about 0.019. So that's a much better indication of how good this model actually is, which is very bad. So on data sets where you have very sparse data or very imbalanced data, the F1 score will give you a really good indication of how well that model is performing. So the loss function that we use for the F1 score is called the Cross-Entropy loss. And it's used because the F1 score metric is not differentiable. So we use the Cross-Entropy loss with a modified prediction. So instead of inputting a 0 or 1 into it, you input like a probability as the S parameter there. So the cross-entropy loss is defined as this negative sum over the number of classes you have, where C is the number of classes, T is the ground truth label for the example, and S is your prediction. And there's a few different types of cross-entropy loss. So we have the three I'm going to talk about now are categorical cross-entropy loss, binary cross-entropy loss, and focal loss. Mainly in this competition, people use focal loss, and I'll kind of explain a little bit about why they decided to just use focal loss. So categorical cross... Oh, a question? Yeah.
I feel like this is something I should know, but I'm not sure. Do you know if I optimize for my cross-entropy loss, have I therefore also optimized my F1 score? Are those equivalent optima or?
Yeah.
Yeah, okay.
So they're basically, the cross-entropy loss is the loss function for F1 score. And so the different versions of cross-entropy loss are just kind of iterations on cross-entropy loss that reflect your F1 score. Categorical cross-entropy loss is a cross-entropy loss function that will apply a softmax function to the loss before calculating the cross-entropy loss, or to the prediction, which makes it pretty well suited for multi-class problems. So multi-class being when you have many classes and only one of them at a time is going to apply to your examples. But unfortunately, the softmax activation function doesn't work very well for multi-label problems where you have multiple labels that apply to an example, because the softmax is going to make it so that the classes aren't independent of each other, and the predictions on each class are going to start to kind of overlap and affect each other, and we don't want that. We want those labels to be independent, so your model is going to be good at predicting those irrespective of each other. So the binary cross-entropy loss and the focal loss use a sigmoid activation function, which means that your classes are going to be evaluated independent of each other for the loss function. And the difference between binary cross-entropy loss and focal loss is that focal loss has this tuning parameter, which, so focal loss was originally introduced by Lin et al. At Facebook, and they claimed that this tuning parameter solves a problem of class imbalance because it forces the loss to focus implicitly on your problematic examples. And it does this here by this tuning parameter gamma. You basically set that, and it's going to change the weight of how a correctly classified example is contributing to the loss. Oh, yeah.
I have a few problems with these loss functions, and what Alexi just asked a few moments ago. So I think there should be, you should be able to plot the loss function and the cross-entropy and see if there's correlation between them, because in some experiments I was doing, some of the loss functions have this binary cross-entropy. Some of them are not compatible if you have this correlation between them, it's not like following each other. So if you're optimizing one, you're not actually optimizing the other. So I noticed that empirically. So I was thinking, do you have a methodological way, or I think it's only by plotting it manually and seeing if there's a correlation. That's the only way you can tell, because sometimes some of those things don't work the same way it's advertised.
Are you talking about like differences between these types of losses?
In relation to Alexi question, so if you're optimizing that one, are you optimizing the F1 score? So then, yeah, I realize there's a lot of differences in those.
Yeah, so for when you're using your F1 score as your metric, then the cross-entropy loss or one of its variations is what you're going to be using to try to achieve the best F1 score.
Yeah, thinking about it, I mean, so in this case, we're not in a multi-class situation so much as we're in sort of like a many kind of one versus all.
Yeah,
kind of.
multi-label.
Yeah, and so, but I think like if you train a model on focal loss instead of binary cross-entropy, you will get a different model. It won't quite be the same, right? It's certainly like the little numbers that come out of it will be definitely different. And I guess you'll have thresholds that you'll play with eventually. But so given that the model will be different, like I guess they aren't quite the same, right? Yeah, for sure. But they're probably both good.
Yeah, so each of the different variations on this loss will essentially give you different models, but they kind of apply to different situations. Like the categorical cross-entropy loss applies to the situation of a multi-class example, and then binary cross-entropy and focal loss apply to examples where you have multi-label. So like you have like the one hot output for the categorical cross-entropy loss and the regular cross-entropy loss. And then with binary cross-entropy loss and focal loss, you typically see those used for these examples where you can have multiple, the output has multiple things turned on, or multiple labels apply to the same example. Or you can just have one, you can still have just one label applying to an example, but you could have more. And so, yeah, this tuning parameter here is basically what makes focal loss really well suited for these multi-label examples that have class imbalance.
To be clear, you can use any of these kinds of loss in this kind of problem. It's just that this one happens to work a lot better.
Mm-hmm. So yeah, the focal loss has the tuning parameter, and it makes it so that you correctly predicted examples contribute less to the loss. And that's basically why we see focal loss used throughout this problem that has a very extreme class imbalance problem. So I'll hand it back to David, and he's going to talk about the different types of image classifiers that we're going to see throughout the solutions and that were used throughout the whole competition.
Yeah. So there was a decent variety in terms of the classifiers that people used in this competition. But one of the things that they all had in common was that they were all convolutional networks. So I wanted to talk a little bit about what the basics of that, well, what a convolution network is and what it does. So if you Google a CNN, then you'll come up with an image that looks a little bit like this. You won't find this one specifically, but you'll find something that looks a little bit like it. So basically what's happening at each of these layers is that the image is being represented as a two-dimensional grid. I mean, in this case, it's technically a three-dimensional grid since we have multiple channels, but it's much easier to visualize if you just think of the two. So if you think of the image as being a two-dimensional array, then what you do is you define a filter, which is that little darker box down the bottom there. And then for each of the pixels that overlap with that filter, you multiply the value in the filter with the value in the pixel. And then you add all those up, and you get a result up in the top here. So you do that for all possible combinations of this filter on this image, and then you get basically a slightly downsampled image as a result of it, although you can actually add things in to keep the image the same size, and most people do. I also wanted to talk a little bit about what the filters themselves actually do. So I've got a few examples of what happens here if you apply like a 3x3 filter to an image. On the left here, we have a picture of Grouse Mountain with a little bit more snow on it than it has at the moment. And if you apply the filters in the middle, you get the images on the right. So the filter at the top is what would be typically referred to as a blurring filter. So you can see that it's got ones in all of the entries. And basically what that means is that the output of this filter is such that the pixel is a linear combination of all of the surrounding pixels, which makes it look, well, blurry. The filter in the middle is an embossing filter. So basically what that does is takes the difference between the pixels to the bottom right and the top left, and then it adds them to the pixel in the center. And what that does is it gives the image an appearance of sticking out of the page a little bit. And those filters aren't the sort of thing that I would expect a CNN to use directly. But the reason I decided to provide them as an example is because they're the sort of filters that you might find on Instagram or something like that, for instance. So if you're using a camera to take pictures and then applying some sort of filter through Instagram or some other app, then basically you're doing the same thing as a CNN does when it's trying to see what's in an image. And the example that I've got the last example that I've got down the bottom is something that might actually be a little bit more useful as far as a CNN is concerned. So this filter is what's known as an edge detector. Basically what it does is it does pretty much exactly what the name describes. It sees whether or not you're at an edge. So the only way that you'll get a non-zero value as an output of that filter is if the image is non-uniform in some way in the area that you're looking at. So if the center pixel differs from the edges in terms of like its intensity, then you're going to get a different value out, which means like all of the white highlighted areas in this image are what's been detected as an edge. And typically, a CNN will function by basically applying a whole bunch of these filters in parallel at the beginning, and those will be there to detect some very basic features like these edges or corners or those sort of things. And then it'll apply more filters later on down the line which identify higher level features, so shapes for instance. And then more filters will be applied on top of that, and those will be used to identify even higher level features like patterns in shapes. So if you're looking at a face, then a nose, eyes, mouth, etc. Now, with all that in mind, everything that I've described so far is applied to all convolutional neural networks, the basic architecture of them. But there are a few refinements that you can do to improve the results that you get in image classification. So one of the most popular ones is ResNet, which stands for residual network. And basically the major difference between ResNets and ordinary CNNs is that they have these skip connections in here. So what these do is they take the output from one layer and feed it directly into the next, to a layer a few steps above it. And the reason that they do this is that the authors noted when they were sort of deriving this that basically when you're trying to classify an image, you're applying a whole bunch of transformations to get from your input as the image to the output, which is all the classes that that image could be. And that's much more likely to be a reliable process if you make a large number of small transformations to the image rather than a small number of large transformations. And by having these skip connections in here, then basically all the network needs to focus on is the difference between the input and the output that it wants to introduce right now. It doesn't need to learn how to replicate the input that it got in with some minor changes. It only needs to focus on the changes that it's making. Yep.
I would like to add some independent intuition about how a ResNet works.
Oh, go
so
ahead.
a ResNet work. So res means residual, and residual means subtracting. So basically, what you try to do is instead of like the usual CNN, what it does is it builds features. So it picks what's on the first layer and it tries to add it until it finds something at the end. What these neural nets tend to do is they subtract things. And the reason why is because you work with a lot more information on the way. So imagine that you have a neural net that you want to do something trivial with it, which is make the output of the neural net to be equal to the input of the neural net. If you use a normal CNN, that's actually hard because you have to build the image again from pieces that you've picked at the beginning. But a residual net, it basically departs from that. It will just make all the layers to not subtract anything, and it will give you that. So now you can imagine that if your solution depends strongly on your input, subtracting things is much better than just adding them and figuring out the different ways you can add them. So that's why I understand they're powerful.
I disagree with the analogy of them subtracting rather than adding, because they do actually add the input to what's output as through the layers. The residual part more refers to the fact that it's only working out a small difference that it's trying to calculate between the original image.
Yeah, but adding something that is negative, it's like subtracting. So it basically will add something to the image such that it would give you something else. So you can see like a subtracting in a way, but it doesn't matter. Adding or subtracting is pretty much the same. It's just that you always keep something to subtract it from or add it to, and that's the original image.
I'm going to try to jump on this as well and try to put in my own intuition, and maybe we can have somebody disagree with me too. So the way I always felt like it made sense to me was that as the model is learning, sometimes some things are easy to learn, right? So there's maybe some classes that it only really needs to do a couple edge detectors and do some simple transformations, and then boom, it's already got the answer. Whereas, and then this would allow that information from like a couple earlier levels or layers to get all the way to the end very quickly. Whereas a normal CNN, it would force it to go through every single transformation. And as Pao said, it's actually pretty difficult to keep the same values as you're going through all these transformations, right?
Yes.
Okay. Maybe, yeah, is that good?
I definitely agree with that. That is the main sort of drive behind these. It's much easier to keep information that's present in the earlier layers if you just feed it indirectly rather than if you are trying to like force the network to like do some transformations and then get the same thing out of it as a result at the end. Kenneth.
So I have one more question. Do you have an intuition for these double convolutions? Why there are two convolutions?
The what, sorry?
Like having two convolutions before the steepnet is like every paper tends to make it two. Do you have an intuition for that? Double convolutions, as they call it.
I don't know exactly why it is two layers rather than just
there is a
one,
name they call it.
but
It's double convolutions. There is a name in literature. I don't really understand the intuition behind it.
Yeah, it's just something that essentially, my understanding was basically that they just tried a few different combinations of like the number of layers between the skip connections, and they found that about two worked fairly well, whereas if you just had it as one, then that didn't work too well. And I mean, my understanding of why that might be the case is just because you haven't actually done much between, like in one layer. You haven't done much in the way of transforming things, so.
And you can also think about it as a way of going deep for cheap, right?
I don't think I've seen it, but I think there might be a somewhat theoretical justification for it to have to be at least two. And the other thing I was going to point out is that in the smaller ResNets, at least the ones from the original paper, it's two, but actually in the larger, it's three. The bottleneck blocks in ResNet 50, 101, and 152 actually have three, so it's actually triple convolution.
Yep.
Although I think they do that, they might do the squeezing thing, I think.
Yeah, they do squeezing in between, so I think it's just because of the different architecture that that it works better. Because essentially, I think two of the transformations are meant to be, two of the layers are sort of meant to represent what one layer would be doing in the original network, right? So it's essentially just two layers, not, it's just expressed as three. If that's confusing enough for you. So ResNet is the first thing that people, well, I wouldn't say the first thing, I guess, but it's one of the basic, very popular sort of architectures that's used for these kinds of problems. And by introducing these skip connections, the original authors found that they could make networks significantly larger, like in terms of the number of layers. So prior to ResNet, it was the standard sort of maximum was somewhere around about 20 layers, I think. I think VGG had 19, and that was the best performing at the time. But with the ResNet sort of architecture, you can go all the way up to 150, which is quite commonly used in these kinds of problems. And in principle, you can go up to 1,000 without losing too much in the way of quality. Although I don't think anybody actually uses anything that large in practice. The next architecture I want to talk about is DenseNet, which is basically a ResNet on steroids. Because what it does is it introduces skip connections basically everywhere that you could have them. So every layer has all of the inputs from all of the previous layers. And the reason the authors decided to do this was that, well, the justification was that basically, like, even in a ResNet, you are still sort of losing some information as you go through the architecture. Because in a ResNet, you sort of add the original values to the transformed values, the ones that have gone through the two convolutions already. Whereas here, what they do is they actually concatenate all of the inputs, which means that literally all the information is preserved. This is obviously a lot more memory intensive. So I don't think DenseNets tend to be as large, but they still do tend to get up to the 100 layer range. Another possible variation on ResNet is the squeeze excitation ResNet. Now, this one does something interesting, because in between each of the layers, it introduces essentially a miniature neural network. So what happens there is that each of the channels gets compressed down into a single value. And then, so for instance, if you had 32 filters in the first layer, then that would get compressed down into a 32 length array. Then they pass those 32 values through like a 2-layer neural network, and they use the outputs of that network as weights for the inputs to the next network. So what that allows the architecture to do is to dynamically alter how much emphasis it's putting on each of the channels based on like some overarching features that it's seeing. Like one example that I like to think of when I'm trying to visualize why you would want to do something like this is that if, for instance, like you've got a very simple image classification problem, like you're just trying to tell the difference between cats and dogs, then one of the features that your network might be trained to look for is pointy ears, because that's obviously a very strong indicator that you've got a cat rather than a dog. So if you can identify with a certain degree of certainty that you've got some pointy ears in there, then you might downweight some of the features that might be associated with dogs and focus instead on some of the features that might be focused on cats. And finally, I'm going to talk about what's definitely the most convoluted of the convolutional networks. So, at least the ones that I'm talking about today anyway, the Inception network. So, there's a lot going on here. This is basically like it can be thought of as a single layer or a single like node, a single block, I guess, in the Inception net architecture. And what's happening is that multiple different filters are being applied to the inputs to generate the outputs. Now, the reason for this is that if you're, once again, trying to classify images, and we go back to our previous example of cats and dogs, then there's a lot of variation in terms of scale that you can have in those images. You could have an image that's like where there's a dog in it and its whole body is in there, but it only occupies a very small portion. It's a long way away from the camera. Or alternatively, you could have an image of a dog which is entirely occupied by the dog's head. So for that reason, there's a large variation of scale in between these kinds of examples that you might have, and you want to be able to draw useful information from both of them. So larger filters, like the 5x5 filter, tend to pull in information from larger range of pixels, which means they're much more useful when you're trying to draw out features from a large area, when you're very zoomed in, essentially. Whereas smaller filters tend to be much more efficient, and they're much better at identifying local features. So, essentially, what they do in the Inception network is apply all of them, just in parallel. So they have like a 1 times 1 filter, a 3 times 3 filter, a 1 times 1 filter, a 3 times 3 filter, and then a 5 times 5 filter, as well as a max pooling layer. They apply all of those sort of in parallel and then concatenate the results to get the output. Yes, Kenneth?
Yeah, so I keep getting confused with the 1 by 1 vertical filters. So what does it do? If it's 1 by 1, is it not identity? Does it do anything? It's like, in signal processing, what does it really mean?
It's 1 by 1 in the dimension of the image, but you've also got like a number of filters, right? So it's actually, let's say you've got 32 filters, then it will be 1 by 1 by 32. So basically it's weighting the pixels in each of the channels and then producing like an output based on that. I know it's a little bit confusing. It's about to get more confusing. So Inception version 2 is basically this. It might not be easy to see from this, but it's basically the same architecture as you have in Inception version 1, except that what they've done is they've replaced the 5 times 5 filters by a 3 times 3, and a 1 times 3 and a 3 times 1. And they've replaced the 3 by 3 by a 1 times 3 and a 3 times 1. And the reason they did this was basically because you can get the same sort of results by applying 2 filters in sequence as you can if you apply 1 large filter, but it's much more computationally efficient to do it this way. So basically, this change in architecture was motivated entirely by computational savings. So like a 3 times, to provide a solid example, a 3 times 1 has 3 parameters in it, and a 1 times 3 obviously has the same, so it's 6 parameters total. Whereas if you had a 3 times 3 filter, that would be 9 parameters, which means that you've reduced the number of parameters in your network by a factor of about a third, which makes it considerably faster when training. I'm not going to go into Inception version 3 or 4. Inception version 3 is very similar to this, except that it's also got like a 7 times 7 filter in it, which has been broken down into smaller filters again. I mean, I could give an entire talk just on the Inception network, but I don't think that would be particularly interesting. So with all that done, I'm going to talk very briefly about our solution. It was a very simple one, and it's placed us at 657th in the competition out of 2,172 participants. We basically just used a ResNet 34 with the 4 channels. So 34 means that it has 34 layers. It's one of the smaller architectures that people use in practice. And we used the full 512 times 512 images as input. We did some very basic sort of data augmentation prior to feeding it into these networks. We basically just did some rotations, some shears, and then some fluctuations in the intensities for the input pixels. That's about it. Some other things that we tried training a CNN from scratch, because we figured that this kind of problem is different enough from the usual image classification that you might want to try a completely different architecture. But that didn't end up being too useful. Yep.
So by implication, your main solution was not trained from scratch.
No.
And
this was a pre-trained ResNet 34.
Pre-trained on 4 channels?
Pre-trained on 3, but the 4th channel was copied over from the red channel. At least in terms of the initial weight. We then did a whole bunch of retraining afterwards.
It's an interesting choice of what to do, so I wanted to know, yeah.
Yeah, fair enough.
Some people chose to initialize it from 0, but they, like other people, did the thing with the red channel, and like if you remember earlier, the red channel and the yellow channel were very similar. So that's sort of a justification for doing that.
And it tended to converge a lot faster if you did that as well, which makes sense because if you start from a non-zero value, then you're much more likely to get a useful result from it. Yep.
You're saying you try to train a basic CNN from scratch. Did you try to train a ResNet 34 from scratch?
Not from scratch, no.
Because
Not too sure what we
that would probably be a lot easier to train than this just a vanilla CNN.
Agreed. Well, easier in terms of more likely to fit the data, yes. In terms of actual CPU time, not so much.
I don't know.
Well,
I'm not saying it's a good idea.
in terms of like, by way of comparison, the CNN, I mean, we were using a fairly basic CNN anyway. We were using this sort of as a baseline to start off with. But it took a few hours to train, whereas the ResNet 34, oh, sorry, I should say it takes us a few hours to train on the 128 times 128 images. That's what we started off with typically. Whereas the ResNet 34 took a couple of days on average, at least with the hardware that we had to throw it at, which wasn't very substantial. The ResNet 34 with the 512 times 512 actually took a couple of weeks for us to train.
So what kind of hardware?
Basically just a CPU. Neither of us had a GPU to throw at this, at least not a useful one. We both have AMD GPUs on our laptops, so.
Okay, in the future, for you guys and for everybody else, there are a couple extra GPUs that some people own in this community. And so if you are in need of a GPU to train something, or if anybody is in a Kaggle competition and they want a GPU to train, I know I have one that's often not in use. So like maybe just make a post in the Slack channel. And I mean, training this on a CPU is just not advisable.
No. That being said, both of us will have access to a useful GPU like fairly soon. He's already built his own computer with one in it, and I'm going back to Australia and I'm going to be picking up my computer, which already has one. So,
Matt, can you elaborate a little bit more on how would that work for people that work with laptops at their home?
Not
like
very
they
well.
would.
Yeah. Well, I mean, like you can easily remote access onto my desktop as long as I think Linux machines can do that pretty easily. I don't know about Windows. I think can do it as well, like
I think you just
using
need
PuTTY.
like a terminal, right?
Yeah, and then you just need to transfer over your script onto my computer and work from kind of in a terminal and run the script, and I should use the GPU. I've helped Stan out before.
Yeah, it worked.
And it worked, yeah. And Kiri is like always on my GPU, but she built her own, so it's now free for other people to use. And it actually gives me great pleasure to know that my GPU is going to good use, right? Because you spend a lot of money on it and you want to use it as much as possible. But I'm only one person.
So we tried a couple of other things as well. Like, as I said, we started off with the 128 times 128 images just because it was faster. But those got significantly worse results. Like, resolution proved to be something that was very important for this challenge. I think the difference between our leaderboard score of like, so we got about 0.44 for the 512 times 512, and I think we got about 0.3 for the 128 times 128. We got slightly better results with the ResNet 101, but that also took about twice as long to train, which meant that we were staring down about a month of training time, about 2 weeks out from the deadline of the competition. So.
I just wanted to ask some questions about the domain of the problem, just to kind of explore that importance of resolution here. Like, so, I mean, like, what do these labels mean exactly? Like, you know what I mean? Like, so I mean, we have some example images here where there are like evidently many cells in the image, right?
Yep.
And so when we have a label, is that label that like there exists some of this fluorescent protein in one of the cells in one of the places, or some of, or all of?
At least one of, yes.
At least one, yeah. But I mean, do you know in practice how as a human, as a lay person that we are, like in this domain, like would we have any hope of actually inspecting these images in any way and drawing a label for it?
I mean, honestly, I wouldn't be able to interpret these images and then be able to identify where it is that the protein was in it. For a start, one of the huge problems is that different cells have extremely different architectures. So like if you're looking at a B cell, then those tend to be very small. They're like a part of the immune system. And most of the time, they just have like the nucleus, which has the DNA in it, and then very little else. They spend most of their time dormant, looking for something to kill, basically. On the other hand, neurons are huge, and they have huge branches, coming out from them. So different structures within cells tend to be in very different places, and some of these locations aren't even present in some cells. So, and they're often not visible just by looking at them. You just have to know, for instance, that this shape of cell corresponds to this kind of cell, and that those kinds of cells tend to have these structures in this part of the cell. So it's not something that would be very easy to do. As I said, you need a specialist to do it, essentially, somebody who knows enough about these kinds of microscope images and these kinds of cells you're looking at.
Okay, and then related questions would be, do you think that these images tend to have all of one type of cell in them, or do you think that they might be kind of a heterogeneous mixture? And then the related question would be, if you broke one of these images into 4 pieces, do you think that each piece would have the same labels as the whole, or do you think it wouldn't? To the last one, because one thing that people did try to do, as a source of data augmentation, was to basically try different crops on the images. And they found that it was very unstable with relation to that. So,
yeah, we'll talk more about that. The 8th place solution mentions that and goes into a bit of detail. But I think for the most part, it was one type of cell in each thing.
So my understanding was that these were cell. I'm not sure if these were all cell cultures, but I think they were all definitely taken in vitro rather than in vivo. So they weren't it wasn't that they cut somebody's cells out and then put them on the microscope to examine them. For a start, to actually make these images in the first place, the first step is to genetically modify the cells that you're interested in, and that's not something that's ethically approved for live humans. So these are all cell cultures that they're looking at, presumably, which means that in cell cultures you typically will have all of the same kind of cell. That was basically what I wanted to talk about with regards to our solution, and I'm going to hand over to Jordan now to talk about some of the better solutions.
Okay.
We have a couple of questions.
So I've got 2 questions actually. In terms of, one is about the architectures and how you choose an architecture. So I tried a ResNet 50 with basically whatever you did, same stuff, and I got something slightly, so I got 0.47.
Okay.
And that made me wonder. So I also briefly tried, because I didn't have much GPU time either, I briefly tried an Inception network. It didn't even seem to converge, so I abandoned that. So my first question is, how do you go about choosing an architecture, or once you've tried one architecture, if you decide that you want to try another one, how do you go about choosing your next architecture? Do we know which ones perform better, or at least once you see your validation loss, how it changes your metric? How do you, did you see in top solutions, did they explain how they decide on what architecture they want to use? And my second question was about the loss function you're using. So my understanding of binary cross-entropy was that you are treating these problems basically as 28 separate binary classification problems. So your network is going to produce 28 numbers, and each of them is either positive or negative for one class, and they're independent of each other. So what did you do, or the top solutions do, when all of those 28 numbers come out negative? So all of your basically numbers are basically close to 0. All of them say, we know that each image has at least one label, but what if all of those 28, because that happened to me for about 10% of my examples, and I had no idea what to do.
So one thing, with regards to the last question, I think one thing that a lot of people did was they played around with the thresholds. So for instance, if for a particular class, you always get results that are somewhere around, at the highest, 0.2 or something like that as an output, then you could set your threshold to be 0.1, for instance, so that everything above 0.1 is considered a positive result for that class. That's not
You're
necessarily the
basically
most...
making your network, you're forcing it to predict something. Is that what you
Essentially,
mean?
that's
Right.
a lot of people did that. I preferred not to play around with that because that seemed a little bit too hacky. To answer your first question, I think it's mostly just experience.
Yeah, in the winning solutions too, we'll talk about a little bit about how people chose those, and what you'll find is that people chose everything. So they use like these big ensembles of multiple models using like pretty much every single network that we talked about and combining the predictions of all of them to come up with some end prediction.
Yeah, the trouble is that this is a sort of solution that they can only really implement because they have so many resources to throw at it. They can try everything and then see what works. I have a hunch as to why your Inception nets didn't train very well. They're very fragile, so the fact that they've broken up into that sort of structure means that like any sort of changes to them can quite easily break them, unfortunately. So they're the sort of method that I would only ever really use with caution. But some people got some good results out of them.
Sorry, can I also add something about GPU time? So I haven't tried it, but I hear that Kaggle provides free NVIDIA K80 GPUs. And also, if you've got a Gmail, then Google gives you $300 of free trial with their ML engine, so they can use whatever GPU they have.
I think it times out after 6 hours. So
Yeah,
you only have 6
the Google
hours
one?
of time.
For Kaggle.
Yeah.
Google, I
Kaggle.
presume, would be different.
Google one
but-
is valid for 1 year.
But
No, I think it like the kernel will just time out after 6 hours, or I think Google's the same, like 6 hours. It'll just
Really?
No, I've actually used the Googles for
thought
like
Google-
a month.
Oh, really?
I assume
Yeah.
Google would
Okay.
be more like AWS.
Yeah, you got like 5 minutes left.
Okay.
Okay.
Enjoy this conversation, but I do want to see the winning solution.
Okay.
I'm going to take just 20 seconds to say, in practice, when you want to select the best architecture for these things, you Google the established trade-offs between performance and flops in models, and you find the like outside curve, and you just pick the biggest thing that you can fit. And there's sometimes little details like an Inception architecture might be a little bit worse, but for the most part, bigger is always better. And if it was better at ImageNet, it'll be better at your problem.
Okay, so I'll try to go through these pretty quickly, and if you have questions about them, you can always ask me after this because we don't have much time left. But the first place solution was very, very interesting because it got like a score that was like 0.02 over the second place solution, which is kind of abnormal. Like usually they're within 0.01 of each other, like the top solutions. So the way they did this was this ensemble model of a DenseNet CNN and this metric learning model, which you can think about this metric learning model if you're familiar with like an SVM or something that uses a nearest neighbor. And what they did was they set a threshold and they said, here's the example, find me the most similar images, and if there's a similar image within my threshold, just take the labels from that and pretty much ignore whatever the CNN gave me. And they were able to improve their score by 0.03 by doing this. So it took them from like I think low 10th place to like first place. So that was pretty significant. And they didn't go into exact details about like how they threw away the CNN prediction because they're actually using this on like the whale prediction competition that's ongoing. So remember that name if you're interested in like actually figuring out their entire ensemble, because they said that they would be posting it after that competition is over. So yeah, that one was pretty interesting to find, and because they had such a significant score over the second place solution. So jumping now to the third place solution, this one we want to talk about because they use this thing called auto augment, which was initially introduced by Google Brain, which was basically your network learns an augmentation policy. So you're not trying to like figure out what the best type of augmentation, like transforming or flipping or whatever it is that you're doing. It just does that for you. You don't have to think about augmenting your data. It'll just learn what's the best augmentation for these types of images. And they mentioned that for the the F1 score threshold that it was really, really sensitive. Like you said, that they were getting many negative examples. And they just kind of matched the threshold that had like, okay, what's going to be the number of positive examples in my validation set compared with like my the training set. So my training set, the number of positive examples in the training set, let's set the threshold so it's about even with the validation set. So that's how they tuned their threshold for their F1 score. And yeah, the models are listed up there that they kind of ensembled together and took predictions from all three of those models. And then the 8th place solution, we're kind of jumping down, but we wanted to talk about this one just to kind of compare how important GPUs are. So they had an ensemble of 14 models. And so for reference, we both use, like our solution and their solution used the 512 by 512 resolution models, but they had 4 1080 Ti GPUs that they were training on, and their 1080 Ti's took the same training time, but were able, as our CPU, like 2 weeks, but they were able to train 14 more models than we were able to train. So that's kind of like shows how significant an increase in computing power is for image classification problems.
And their models were significantly larger than ours as well.
Mm-hmm. Yeah. So, yeah, they had just a whole whack of models. I'm not sure that that they needed them all, but that's what they did. They also tried to use some complex augmentations, like using a generative adversarial network to generate samples, but they found that it didn't help. And like, just to kind of theorize or some intuition about why that didn't work is because these scores were so low for all these models, like they were all under 0.6. If we're thinking about what GAN is doing, is kind of giving your or it's trying to fool your model into thinking like this is a real sample. And if your model doesn't really know what a real sample is supposed to look like anyways, like it's less than 0.6 or 60% confident, I guess you could say, and this is a real sample, then it's going to be pretty easy to fool, and you're going to be providing it with low quality data. And the last thing to mention about this is the cropping. So why you can't crop these images is because there are certain proteins or certain organelles in the cell, like the cytokinetic bridge was one of the examples, that occur once per cell. So depending on your crop, you could be cropping out all the relevant information to what the label is for this specific example. And then what we want you guys to take away from the presentation and what we learned. So F1 score, it's useful for these unbalanced data sets. And the loss function for F1 score is cross-entropy, and there's those 3 types that are used for different examples. So you have the categorical, which uses the softmax activation, and that's used for the multi-class problems, so like a one-hot vector as your output, and then binary cross-entropy loss and focal loss are used for the multi-label outputs where you have multiple multi-hot vector as your output, I guess you could say. Ensemble models were very, very popular in this competition. So not just relying on one model as the output, but taking a bunch of outputs and coming up with your prediction by combining those together. And then the winning solution, which was really interesting, was the ensemble of the CNN plus the metric learning model, which kind of just threw away those CNN labels based on similar examples and just stealing the labels from those.
Going into this competition, one of the things that I was also looking for was some practical experience with actually training networks and trying to solve a problem. And that's one thing that Kaggle is really good at. So there's a lot of things that you only really learn by building the networks yourself and trying to get them to work. We also learned a lot about some more, I don't know, esoteric topics about how you would solve these problems. So like, the first thing that I would definitely recommend if you're ever looking at a Kaggle competition is to go to the kernels and to like look at the discussion as well, because Like our solution was basically one of the kernels that we'd taken, and made a few minor modifications to it to improve the results. And the first place solution was actually the same. They used the same kernel as we did. So it's very easy to get some good results, and there are often some very good kernels on the Kaggle website that you can use to as a baseline for building your solution out of.
Mm-hmm. And retroactively looking at some of the discussions after it had ended, you could see oh, this is the 5th place solution, and they're in this discussion talking about oh, I'm going to try this, and you can see that all these top solution people are in those discussions, contributing, saying what they're going to do. So you can look at these top Kagglers and kind of try to follow what they're doing as well.
Mm. We also learned quite a lot just by preparing for this presentation. We had to read a lot about the winning solutions, as well as some of the models that they decided to use and how they differed from what we were familiar with. So we can definitely recommend this, just presenting as an avenue for learning, even if you don't have time to participate in a competition itself.
Good job.